Hi, everyone. I'm Molly Monge, and I am a postbac in the SADU lab in NHGRI. Many molecular biology experiments are limited by the number of experimental replicates that you can generate and the conditions by which we perform these experiments. It can be difficult to generate experimental replicates and also difficult to test all these in one setting. As we know, most experiments are performed over the course of several days, and confounding factors based on the conditions that we perform these experiments in can influence our data. Why do we perform experiments like this? And why is there no method to more efficiently generate replicates and perform experiments more in a pooled format? My project is trying to answer these questions as I am developing a framework that could be applied to many experiments to limit these concerns. I'm developing this framework in the context of the nucleotide excision repair factor 1 complex pictured here on my screen. The NER pathway is a DNA repair pathway conserved amongst several organisms, and it's a way for them to respond to DNA damage. The 3 yeast genes in this complex are circled here in red, RAD1, RAD10, and RAD14. We currently lack a good understanding about how these proteins interact, and my project is interested in better understanding these interactions, as well as understanding the degree to which these different genes can be interchanged with human homologs. The NER complex is implicated in many different diseases in humans, so understanding how these proteins function and interact is of key concern. Using this complex of proteins, we will generate plasmids that contain all possible combinations of the NER genes, those being the yeast and human homologs, as well as non-coding DNA that serves as a negative control or a knockout. Each of these genes will be tagged with a unique set of barcodes that allows us to track them throughout the experiment and to generate many experimental replicates. After generating all possible 27 combinations, we will transform these plasmids into a yeast strain that has the RAD genes knocked out. We will treat these yeast with a genotoxic drug called methylmethylsulfonate, which induces DNA damage and triggers this repair pathway. We can use Illumina sequencing to understand how the different combinations of the NER genes respond to this damage by measuring barcode abundance before and after exposure. We would expect that barcodes that appear at a high abundance after MMS exposure could respond to the DNA damage and survive, while those that drop out of the pool struggle to survive the MMS exposure and represent a non-functional or less functional version of the NER complex. By performing this experiment with trackable barcodes and in a pool, we limit confounding factors, and we're generating thousands of experimental replicates. We imagine that many other biology experiments would benefit from a framework like this. Our experimental approach should give scientists greater statistical power to draw conclusions about their data. Thank you for taking the time to listen to my talk.